A very good evening to all of you. How are you? How is your preparation going on? Today we are going to do biochemistry. This biochemistry is very important as far as NEET PG is concerned. Why? 16 questions are fixed in NEET PG and about 8 to 12 questions in INSET from biochemistry. And it is a good subject if you have your basics clear. You can answer many questions of medicine and pediatrics also due to a good grip in biochemistry. So let's begin with the first question. ApoB48 is a remnant of ApoB48 is a remnant of A. RNA editing, B. RNA interference, C. RNA splicing, D. DNA translocation. What happens in liver? ApoB100 gene gives rise to ApoB100 protein, but ApoB48 protein is made in intestinal cell from the gene encoding ApoB100 by the process of RNA editing, which is also known as differential RNA processing or chemical modification of RNA. This is post-transcriptional modification. RNA splicing is also a post-transcriptional modification, but this is removal of introns and not editing. RNA interference is a silencing technique where the gene is silenced by not letting the product of gene form. Translocation occurs during translation. This movement of ribosome over mRNA. It is RNA editing. Hepcidin decreases iron absorption by inhibiting A. Divalent metal transporter, B. Hephestin, C. Ferroportin, D. Transferrin. It is ferroportin. So correct answer here is ferroportin. What is that? Hepcidin. Hepcidin regulates iron metabolism. It mainly regulates iron entry in the circulation. By inhibiting ferroportin, hepcidin reduces dietary iron absorption. The iron released from macrophage is also reduced by ferroportin inhibition. So increased hepcidin activity is responsible for reduced iron availability. Increased hepcidin will lead to anemia. Decreased hepcidin will lead to iron overload. Hepcidin synthesis and secretion by liver is controlled by iron stores within the macrophage, inflammation, hypoxia, and erythropoiesis. Divalent metal transporter absorbs divalent metal ions from intestine, especially ferrous. Hephestin involved in homeostasis of iron and copper. Transferrin is a protein which is responsible for the transport of iron. So the correct answer is C, ferroportin. Which of the following has highest glycemic index? A. Glucose, B. Fructose, C. Sucrose, D. Sorbitol. It is glucose. GLUT5 is a transporter for A. Galactose, B. Fructose, C. Mannose, D. Glucose. So what do you think is the correct answer? It is fructose. Go to the description. A link is there. Just click on the link in the description. You will reach a WhatsApp group. Stay in that WhatsApp group. You will get the ultimate NEET PG breakthrough blueprint masterclass. It is a 3-day masterclass covering all the 19 subjects in the most holistic way possible, interlinking the subjects. And it will improve your score in your NEET PG and INSET. Which of the following is insulin dependent? A. GLUT1, B. GLUT2, C. GLUT3, D. GLUT4. It is GLUT4. Cause of HHH syndrome: hyperornithemia, hyperammonemia, homocystinuria. Is A. Deficiency of ornithine transcarbamylase, B. Defect in ornithine transporter, C. Deficiency of aminotransferase, D. All of the above. It is defect in ornithine transporter. Defect in ornithine transporter leads to HHH syndrome, that is hyperornithemia, hyperammonemia, homocystinuria. This prevents ornithine transport into mitochondria and hence if in the cytoplasm, and its reduced ability to clear carbamyl phosphate and ammonia. Mitochondrial carbamyl phosphate carbamylase lysine to leading to homocystinuria. So the correct answer here is defect in ornithine transport. Which method is used to separate a mixture of lipids? A. Isoelectric focusing. B. pH. C. Chromatography. D. Electrophoresis. It is chromatography. Wrinkled paper appearance of macrophage in bone marrow aspirate is seen in A. Tay-Sachs disease, B. Gaucher's disease, Niemann-Pick disease, and leukodystrophy. It is Gaucher's disease. Wrinkled paper or crumpled tissue appearance of macrophage in bone marrow is seen in Gaucher's disease. In CRISPR-Cas9 system, which repair mechanism is used for genome editing? A. Homologous repair. A. Homologous repair. B. Non-homologous end repair. C. Mismatch repair. D. Nucleotide excision repair. It is non-homologous end repair. So the correct answer here is non-homologous end repair. Rapport loop brick cycle shifts oxygen dissociation to curve to right because of A23-DPG, B13-BPG, 3-phosphoglycerate, 1,6-fructose biphosphate. It is 2,3-DPG. Hot flashes are experienced as a result of increased noradrenaline, decreased estrogen, increased noradrenaline, decreased estrogen, increased noradrenaline, and increased estrogen. It is increased noradrenaline and decreased estrogen. Restriction fragment and polymorphism is used for A. Analysis of DNA fragment, B. DNA estimation, synthesis of nucleic acid detecting. It is analysis of DNA fragment. Thermogenic food is which of the following? A. High protein diet, high carbohydrate diet, high fat diet, does not depend on macronutrient. It is high protein diet. Ketone bodies are not utilized by A. Brain, B. RBC, C. Heart, D. Skeletal muscle. It is RBC. Enzyme deficient in hyperammonemia type 1 is ornithine diacarbamylase, arginase, carbamyl phosphate synthase 1, arginosaccharide synthase. It is carbamyl phosphate synthase 1. This hyperammonemia is a metabolic disturbance characterized by excess of ammonia in the blood. It is a dangerous condition and may lead to brain injury and death. It may be primary or secondary. This usually occurs in urea cycle disorder. Deficiency of any of the enzyme of urea cycle leads to ammonia accumulation. Type of mutation seen in sickle cell anemia is A. Insertion, B. Deletion, C. Point mutation, frame shift mutation. It's point mutation. Which replacement of amino acid does not alter its normal function? Glutamine replaced by arginine, glutamine replaced by alanine, glutamine replaced by glutamate, glutamine replaced by aspargine. It is glutamine replaced by asparginase. The clinical feature given below occur in deficiency of A. Tryptophan, B. Phenylalanine, C. Cysteine, D. Glutamine. It is tryptophan. Which of the following binds mRNA with ribosome in eukaryotes? A. Poly, A. tail, B. tRNA, tRNA, 7-methyl guanylyl cap, Shine diagonal sequence. It's 7-methyl guanylyl cap. Which of the following is anaplerotic reaction? Conversion of pyruvate to lactic acid, conversion of pyruvate to oxaloacetate, pyruvate to acetyl CoA, conversion of pyruvate to acetyl. It is conversion of pyruvate to oxaloacetate. Which of the following group of protein assist in folding of other proteins? A. Proteases, B. Proteasome, C. Template, D. Chaperone. It is chaperone. Chaperones are protein which assist in protein folding. They are located in the endoplasmic reticulum. They mediate post-translational assembly of protein complex. So it is chaperones. Myoglobin has which structure? Primary, secondary, tertiary. Primary and secondary. Primary, secondary, tertiary, quad. Primary and secondary. It is primary, secondary, and tertiary. Myoglobin has only one chain, so it does not have quaternary structure. Monomeric proteins do not contain quaternary structure. Best investigation for metabolic disorder is A. Western blot, B. Tandem mass spectrometry, B. CPCR, D. Cell electrophoresis. It is tandem mass spectrometry. Separation of protein by their mass is caused by gel filtration chromatography, ion exchange chromatography, isoelectric focusing, epitative chromatography. It is gel filtration chromatography.
ಆತ್ಮೀಯ ಬಿಎಸ್ಎನ್ಎಲ್ ಗ್ರಾಹಕರೇ, ದೂರಸಂಪರ್ಕ ಇಲಾಖೆಯ ಸೂಚನೆಗಳ ಪ್ರಕಾರ